This I found this book like a PDF. It has antibody structure and function. Antibodies, a central part of humoral immunity, have increasingly become a dominant class of biotherapeutics in clinical development and are approved for use in patients as with any and are approved for use in patients. As with any successful endeavor, the history of monoclonal antibody therapeutics benefited from the pioneering work of many, such as Paul Ehrlich, who in the late 19th century demonstrated that serum component had the ability to protect the host by passive vaccination. The seminal invention of monoclonal antibody generation using hybridoma technology by Kohler and Milstein, and the advent of recombinant technologies that sought to reduce the marine content in therapeutic antibodies. During the process of generation of humoral immunity, the B cell receptor is formed by recombination between variable diversity and joining exons which define the antigen recognition element. This is combined with an immunoglobulin constant domain element that defines the isotype of the molecule sequences for these VDJ and constant domain genes for disparate organisms can be found through the International Immunogenetics Information System. The different Ig subtypes are presented at different points during B-cell maturation. For instance, all naive B-cell express IgM and IgD, with IgM being the first secreted molecule. As the B cells mature and undergo class switching, a majority of them secrete either IgG or IgA, which are the most abundant class of Ig in plasma. Characteristics like high neutralizing and recruitment of effector mechanism, high affinity, and long resident half-life in plasma make the IgG isotype an ideal candidate for generation of therapeutics antibodies. Within the IgG isotype, there are four subtypes with different properties. Most of the current marketed IgGs are the subtype Ig1, heavy chain constant gene, approximate molecular weight kDa, mean serum level, half-life in serum, ADCC, CDC, number of disulfides in hinge, etc. The ability of antibodies to recognize their antigens with execute specificity and high affinity makes them an attractive class of molecules to bind extracellular targets and generate a desired pharmacological effect. Antibodies also benefit from their ability to harness an active salvage pathway mediated by the neonatal Fc receptor, thereby enhancing their pharmacokinetics, lifespan, and mitigating the need for frequent dosing. The antibodies and antibody derivatives approved in the US and the European Union span a wide range of therapeutic areas including oncology, autoimmunity, ophthalmology, and transplant rejection. They also harness disparate modes of action like blockade of ligand binding and subsequent signaling, and receptor and signal activation, which target effector functions. ADCC and complement dependent cytotoxicity and delivery of cytotoxic payload. Antibodies are generated by the assembly of two heavy chains and two light chains to produce two antigen binding sites and a single constant domain region. The constant domain sequence in the heavy chain designates the subtype. The light chains can belong to two families, with the most current with most of the currently marketed antibodies belonging to the K family. The antigen binding regions can be derived by proteolytic cleavage of the antibody to generate antigen binding fragment and the constant fragment. The Fab comprises the variable region employing immunization followed by recovery of functional clones, either by hybridoma technology or more recently by recombinant cloning of variable domain from previously isolated B cells, displaying and expressing the desired antigen binding characteristic. There are several variations of these approaches. The first approach includes the immunization of transgenic animals expressing This approach requires the use of a genotype-phenotype linkage strategy (Fab, heavy chain, light chain, hinge). Topologically, the IgG is composed of two heavy chains and two light chains with total molecular weight of approximately 150 kDa. Each heavy chain is composed of four domains. The variable domain, the right chain is composed of variable domain and constant domain. All domains in the IgG are members of the Ig-like domain family and share a common Greek key beta sandwich structure with conserved intradomain disulfide bonds. The right chain pairs up with the heavy chain's domain to form the Fab fragment, while the heavy chain CH2, CH3 domains dimerize with additional heavy chain. Fc domain is connected to the Fab domain via flexible hinged region that contains several disulfide bridges that covalently link the two he- heavy chain together. The right chain and heavy chain are also connected by one disulfide bridge, but the connectivity differs among the IgG subclasses. The overall structure of IgG resembles a Y shape, with the Fc region forming the base, while the two Fab domains are available for binding. Studies have shown that in solution, the Fab domains can adopt a variety of conformations with regard to the Fc region. While the Fab region of an antibody is responsible for binding and specificity to a given target, the Fc region has many important functions outside its role as a structural scaffold. The Fc region is responsible for the long life is responsible for the long half-life of antibodies as well as for their effector function, including ADCC, CDC, and uh, the long half-life of human IgGs relative to other serum proteins is a consequence of the pH-dependent interaction with the FcRn, the endosome. Ah! Fc region is also responsible for binding to bacterial protein A and protein G, which are commonly used for purification of Fc-containing proteins. Although protein A binds to Fc mainly through hydrophobic interaction and protein G through charge and polar interaction, proteins A and G bind to a similar site on Fc domain and compete with each other. Interestingly, the binding occurs between the CH2-CH3 domain of the Fc and largely overlaps with the FcRn binding site. One major characteristic of IgG, which differs from other IgG isotypes and most of the other serum protein, is its long serum half-life. Typically, serum protein and other IgG isotypes have half-life of less than 1 week. For example, FcRn, which is structurally homologous to MHC class 1 molecule, is a heterodimer consisting of 50 kDa transmembrane A chain and 12 which is required for the FcRn expression. FcRn mostly reside intracellularly but can be exposed to the extracellular environment. FcRNs of multiple mammalian species have been cloned, and functional expression of FcRn has been reported in mammals like the rat, mouse, rabbit, sheep. A number of studies have demonstrated that balancing the affinity kinetic improvement of Fc variant at pH 6.0 and pH 7.4 is critical for engineering variant with improved half-life. The most compelling data are obtained from association of clinical outcomes with functionally relevant receptor polymorphism (PCR)